Hello everyone, I'm Sham Asad. Today I'm going to present a research titled Spike Proteins and Their Role in the Entry and Fusion of the Coronavirus with the Host Cell, supervised by Dr. Nidal Hassan, and we're going to start it with the introduction. Coronaviruses belong to the family Coronaviridae in the order Nidovirales. They can be classified into four genera: alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. Among them, alpha and beta coronaviruses infect mammals. The etiological agent of COVID-19 is a new member of the Coronaviridae family that is closely related to severe acute respiratory syndrome coronavirus. Coronavirus consists of spike glycoprotein, M protein, hemagglutinin esterase dimer, envelope, RNA and N protein, and E protein. Coronaviruses are enveloped positive sense RNA viruses. Enveloped coronaviruses entering host cells and initiating infection are achieved through the fusion of viral and cellular membranes. Membrane fusion is mediated by its spike protein. The statement of problem. What caused the great harmful effect of coronaviruses on the alveoli in the lungs? And to answer this question, we have to mention the outlines, which are coronavirus spike proteins, hemagglutinin esterase protein, life cycle of coronavirus, and then we have the conclusion. Coronavirus spike proteins. Coronaviruses demonstrate a complex pattern for receptor recognition. For example, the alpha coronavirus, H coronavirus, and L63 recognizes a zinc peptidase angiotensin converting enzyme 2. Moreover, the alpha coronaviruses TGEV, PEDV, and PRCV recognize another zinc peptidase, which is aminopeptidase N. Similarly, beta coronaviruses recognize different receptors. For example, MERS coronavirus and HKU4 recognize a serine peptidase, dipeptidyl peptidase 4. MHV recognizes a cell adhesion molecule, carcinoembryonic antigen-related cell adhesion molecule 1. B coronavirus and OC43 recognize sugar. Other than their role in viral attachment, these coronavirus receptors have their own physiological functions. Two major domains in coronavirus S1, N-terminal domain S1-NTD and C-terminal domain S1-CTD have been identified. One or both of these S1 domains potentially bind receptors and function as the receptor binding domain RBD. S1-NTDs are responsible for binding sugar, with the only known exception being beta coronavirus MHV S1-NTD that recognizes a protein receptor CEA-CAM1. S1-CTDs are responsible for recognizing protein receptors ACE2, APN, and DPP4. The structure of beta coronavirus SARS coronavirus S1-CTD complex with the human ACE2 provided the first atomic view of coronavirus S1. Hemagglutinin esterase protein. To aid viral release from infected cells, some coronaviruses would also evolve a hemagglutinin esterase protein as a receptor-destroying enzyme. Hemagglutinin esterase protein is a glycoprotein that certain enveloped viruses possess and use as invading mechanism. And now let's move on to the life cycle of coronavirus. After the COVID-19 enters the body, it needs a special receptor to help it enter the cell. This receptor is called angiotensin converting enzyme 2, and it is found on the surface of the alveolar cells in the lungs. The life cycle of SARS coronavirus 2 or COVID-19 begins with membrane fusion occurring at the plasma membrane or within acidified endosomes after endocytosis, which is mediated by conformational changes in the acid-glycoprotein triggered by angiotensin converting enzyme 2 binding. Following viral entry, SARS coronavirus 2 releases its genomic RNA into the host cell cytoplasm. Genome RNA is first translated into viral replicase polyproteins, which are further cleaved by viral proteases into a total of 16 non-structural proteins. A replication transcription complex RTC is formed based on many of these non-structural proteins. In process of genome replication and transcription mediated by RTC, the negative sense genomic RNA is synthesized and used as a template to produce positive sense genomic RNA and subgenomic RNAs. In the rough endoplasmic reticulum and Golgi complex, the SARS coronavirus 2 glycoprotein is subjected to co-translational and post-translational processing. The N protein is subsequently associated with the positive sense genomic RNA to become a nucleoprotein complex or nucleocapsid, which together with S, M, and E proteins as well as other viral proteins is further assembled and followed by budding into the lumen of the endoplasmic reticulum Golgi intermediate compartment to form mature variants. Finally, the mature virions are released from the host cell waiting for a new life cycle to start. There are three types of alveolar cells. Type 1 it is responsible for gas exchange with blood vessels. The second type is responsible for the production of the surfactant, which is a mixture of proteins and lipids that reduce the surface tension of the alveoli. Type 3 immune cells or dust cells, and they are macrophages. The ACE2 is on the type 2 of these cells. Because the life cycle of COVID-19 is a lytic cycle, the type 2 alveolar cells die, and they release substances called specific inflammatory mediators. These substances stimulate the existing immune cells macrophages, and when they are stimulated, they secrete three immune substances called cytokines, which are interleukin 1, interleukin 6, and the tumor necrosis factor. When these three substances reach the bloodstream, they cause the symptoms associated with infection with the coronavirus. The conclusion. SARS coronavirus 2 is a highly contagious pathogen that continues to spread quickly around the globe, causing COVID-19 to be one of the worst pandemics in recorded history. A safe and effective vaccine represents one of the best ways to reduce or eliminate the COVID-19 pandemic. Vaccines like AstraZeneca, Moderna, and Pfizer-BioNTech vaccines have contributed to reducing symptoms and limiting the spread of the disease. But the mutations that occur to the virus permanently increase the doubt about the survival and the long-term effectiveness of vaccines against the virus. The virus is still in a non-stop development as long as it can clone itself. These are some of the references that were used in this research. And thank you for your attention.